Okay, Year 11s. Today we are going to be looking at Chapter 1.3 and Chapter 1.4. So Chapter 1.3 is on cell division, Chapter 1.4 is on cellular requirements. Chapter 1.3 goes from slides 47 to 53, so we'll start there. Cell division is really the true purpose of the cell. It's why the cell actually exists. If cells weren't able to divide, then they would only exist for one generation and there would be no living organisms left. So all life depends on this constant reproduction. Because we have different cell types, and as we've described, they've got lots of different ways that they live, they also have different ways of reproduction. And there are two main ways. The first, prokaryotes, is more simple, it's binary fission, and then the second, in mitosis, is a bit more complicated. That's what we do in eukaryotes. So eukaryotes are mitosis, prokaryotes is binary fission. Now with binary fission, it is quite a simple process. We'll just quickly do the diagram for that now. So binary fission, we start with our cell wall and our cell membrane. So this is our bacteria, and then within here we have our bacterial chromosome. So very quickly we need to do our labels for that. So here is our cell wall, blue is our cell membrane, and then in here is our chromosome. Now, the very first step of any kind of cell division is the duplication of the chromosome. So that's what we have to do here. Step one, duplicate the chromosome. Very, very simple. Then in step two, what the bacterial cell does is it begins to elongate. So it stretches itself out. You need to draw the bacterial chromosome duplicating. That doesn't necessarily cover the, go around the first chromosome, it's just because we can't draw in 3 dimensions. But we have our elongated bacterial cell with its cell wall and cell membrane just as we had before, and because the chromosome is attached to the cell membrane at a specific point, as it elongates, it pulls the chromosome apart. So step two. Cellular elongation separates the chromosomes. And then in step three, we have our 2 new cells. Cell wall, cell membrane, and our duplicated chromosomes. So it pinches in to form 2 daughter cells. Now, there is a slight weakness with this. This is asexual reproduction. And asexual reproduction produces clones, which means there's no genetic diversity. So very simplistically, if you throw some antibiotics at bacteria that's duplicated via this, if it kills it, it's going to kill all of them. If it doesn't, it's going to resist the entire bacteria. But the problem is, antibiotic resistance is relatively rare. So this is a significant problem. Bacteria have actually developed a way around it. So this is our binary fission, asexual process, but bacteria also have a mechanism to increase the amount of variation that they actually are able to achieve. Genetic variation, that is. The way that they do this is by this process called bacterial conjugation. Now, bacterial conjugation, we'll put that as our next heading. And this time we won't draw the cell membrane with the cell wall, just because it's time consuming. So we've got our, once again, got our chromosomes, and this time one cell has a second structure. So the second structure is called a plasmid. Again, we'll just call that the cell boundary. So it's made up of the cell membrane and the cell wall. And then here is our bacterial chromosome. What happens here is that the two bacteria, for whatever reason, they form this bridging structure. And this bridging structure then allows the bacterial plasmid to copy itself and reform in the second cell, so that we have a copy of the plasmid in each bacteria. This means that if that plasmid carries the gene for, say, bacterial antibiotic resistance, then both of those bacterial cells are now going to be resistant to that particular antibiotic. So it's a way of increasing that genetic variation, getting over that issue of asexual reproduction, producing only clones. The other thing that we consider here, when we're looking at bacterial reproduction, or the production of new bacterial cells, is how fast it happens. Asexual reproduction is incredibly rapid. So we've got this binary fission process, which is an asexual process, and it creates an explosion of numbers of cells. We've talked about our variation here with bacterial conjugation, so we'll get rid of that for now. Now we need to talk about grafting growth. So, grafting bacterial growth. We've got our y-axis, our x-axis. On the y-axis, we're just going to have number of cells, and down here we're going to have time, and just for the sake of it, we'll throw in some units as hours, and the units of cells, we'll call that colony forming units, CFUs. What we have is this very, very simple shape. Start at the bottom, we get an exponential growth, stationary phase, and then a death phase. So we'll shorthand that exponential growth phase, stationary phase, and then a death phase. And the exponential growth phase is characterized by a higher rate of reproduction than death. Cells still die, but it's just that there are far more cells reproducing than dying. At the stationary phase, the rate of reproduction versus the rate of death are equal, and in death phase, the rate of reproduction is far less than the rate of death. So we have this very simple way that we can describe it. So for the death phase, rate of reproduction is less than rate of death. Here the rate of reproduction equals rate of death, and in here the rate of reproduction is greater than rate of death. There's a whole variety of different reasons, and we're going to come to that when we get to 1.4 when we're looking at cellular requirements. The other thing to consider with bacteria, when they're dividing, often we observe bacteria dividing artificially, in vitro, which means in glass, outside of a normal situation. When this happens, particularly on agar plates, which we'll use quite a bit in our lessons or in our practicals, on agar plates they form in colonies, which is almost a unique situation. It occurs very rarely in nature. And it's effectively where a single bacteria or a small group of bacteria give rise to a very large population. We call this a colony because it comes from one colony forming unit, or we assume it's one bacterial cell. And we'll use that quite a lot when we're getting into bacteria and investigating bacteria. So become a practical skill we'll develop, this identification of colonies. So that's bacteria. Binary fission, bacterial conjugation, this rate of reproduction equaling or greater than or less than rate of death when we're graphing bacterial growth, and this understanding of colonies. That's mitosis, that's cell division for bacteria. When we get to cell division for eukaryotes, we have this new concept of mitosis, or not new for most of you hopefully, because you will have done year 10 biology, but new at least in the terms of this particular course. So we'll have our next heading, which is mitosis, and I'll just put next to that eukaryotic division. Now, the other thing that we've got to consider is why reproduction, or why cell division happens. For bacteria, it's only ever reproductive. They don't need to repair, they're only one cell. But because eukaryotes are multicellular organisms, we need to grow, we need to repair, and we need to undergo reproduction as well. We need to be able to pass on our genes. Now, some eukaryotic organisms undergo asexual reproduction, which is a similar sort of process to what bacteria do, and that we end up with clones. But a lot of them undergo sexual reproduction. So growth and repair are exclusively mitosis. Every single time we have growth, or every single time we have repair of an organism, we need those cells to be genetically identical. Whereas for reproduction, if it's an asexual reproducer, it's going to be mitosis. If it's a sexual reproducer, it's going to be meiosis. And we're not particularly concerned about exactly the mechanisms there. But mitosis, the actual period of cell division, is comparatively short. It's a short part of the eukaryotic cell cycle. By far the biggest part is interphase, the phase between, or the part between each part of, or each phase of mitosis, each situation, or each effort of mitosis. So interphase is by far the largest. But because we're primarily concerned with mitosis, we're just going to focus on that. So we're going to draw 4 stages. At the first 3 stages, we're going to have 1 cell, and at the last stage, we're going to have 2. So here we have our chromosomes, and just to make life easier, we're going to draw 2. When we get to our next stage, those chromosomes meet at the equator, and these special little structures form called spindle fibers. We'll label these in a moment. Those spindle fibers then contract in the 3rd stage. And that contraction pulls apart the chromosomes. And then in the very final stage, we have our cells just about at rest again, ready to start the process again. We'll get our labels happening now. So this here we have is our chromosome, and it's duplicated. Here we have our spindle fiber. And then over here we have a nucleus, and we might as well, just for the sake of it, draw our line here to our unduplicated chromosome. Now, each of these steps has a specific label. We call it something specific. So over here, we have what we call prophase. Here we have metaphase. Anaphase, where the chromosomes are separated, and then we've condensed these last 2 phases down, so it's telophase and cytokinesis, and that's the basic process. We've got chapter 1.3 questions you need to do based on that. Those stages are obviously, we've simplified them, focused predominantly on the drawings. You need to be able to identify each stage. For mitosis, you need to be able to explain what's going on. So whether you do that using a diagram and then explain in words, or whether you do some research on it for yourself, that's up to you.